Our final speaker of this morning's session is Ben Stafford. He's a scientist within the particle characterization team at Intertek Melbourne. He's been with them for 2 years. Ben has extensive experience in method development and validation for techniques that fall under the particle characterization umbrella for inhaled and nasal drugs, such as particle droplet sizing via laser diffraction, morphology-directed Raman spectroscopy, and spray pattern plume geometry. He also has experience in device product characterization studies and takes part in research activities exploring new analytical capabilities. Prior to joining Intertek, Ben graduated from Durham University with a first-class MSc in Natural Sciences, which included a research project in characterizing pharmaceutical co-crystals via solid-state NMR spectroscopy. So welcome, Ben.
Yeah, thank you. So I'm from Intertek's Melbourne site near Cambridge, and amongst other things, we are Intertek's Centre for Excellence with inhaled and nasal drug products. And so today we're going to be going through some generics and some in vitro bioequivalence. Then we're going to look at some morphologically directed Raman spectroscopy and some of the challenges that that brings. Then we're going to look at some advanced optimization of MDRS methods. And finally, we're going to tie it all together with a new study regime proposal for some MDRS-based IVBEs. So firstly, for a bit of context, a generic drug is a reproduction of an off-patent drug which is already on the market. And so a generics manufacturer doesn't need to put their product through full clinical trials. They do, however, have to prove that their product is bioequivalent to a reference-listed drug, the one that is already on the market. So conventionally, a bioequivalence study is made up of some in vitro lab-based testing, a PK study, and a clinical endpoint study. And so these are all comparative tests. You perform the same suite of testing on your generic and on the RLD, and if the results are sufficiently similar, you then prove that the generic is suitable to go to market. Increasingly, however, some regulators are offering more in vitro alternatives to those clinical endpoint studies. And so those are primarily through technological advancements, where depending on the product, an advanced analytical technique will be used to characterize a certain key attribute of the drug, essentially making the clinical study redundant. And so bypassing a clinical study has obvious advantages, both financial and time-wise, for the generics manufacturer. And that gives us the concept of an in vitro bioequivalence study, or an IVBE. Very recently, however, so in May of this year, the FDA released some updated guidance for memetazone furoate, fluticasone furoate, and fluticasone propionate nasal sprays, which for the first time are potentially offering fully in vitro only pathways to the market. And so if you're wanting to make a generic of one of these products, you have to do all of these tests, which I'm not going to go through each one individually, but the idea is you do all of these and you may be able to bypass any clinical studies whatsoever. I've highlighted at the end the drug particle size distribution, which was one of the primary updates of that guidance and is also what we're going to be looking at further now. So as was kind of mentioned in the previous talk, and as I'm sure many of you are aware, so for inhaled and nasal drugs, the particle size will directly affect the rate and the extent of the dissolution and the overall PK nature of the product. And so morphologically directed Raman spectroscopy, or MDRS, is a technique which will selectively measure the particle size distribution of a species within a drug blend or suspension. So that's as opposed to something such as laser diffraction, which will measure one PSD of the entire blend. MDRS will be able to pick out the PSD of one species within that mixture. And so to do that, firstly, when you prepare your sample, you want a static monolayer of your particles. So here we have a nasal suspension imaged under a microscope. Then those particles are all imaged and sized via light-based microscopy. So that's the morphologically part of the analysis. And then the MDRS is added on top, where a set of those particles are chemically identified via Raman spectroscopy. And so that provides the selective nature of the analysis. So MDRS is incredibly powerful. However, each Raman spectrum takes at least 10 seconds to acquire. And it can take longer, and that is intrinsic to the species you're looking at. We can't control that. To build a good PSD, you need hundreds, however, often in practice, thousands of Raman spectra. And then if you add all of those together, in practice, what that means is you're only analyzing one sample per day, per system. For an IVBE, you require a minimum of 60 samples. So you're looking at 3 generic batches, 3 reference batches, 10 samples per batch. It could be more depending on the product. Add all of that together, and you've got months worth of analysis time, and so MDRS is comparatively very costly. Still preferable to a clinical study, but compared to one of the previous tests we looked at on the previous slide, so say plume geometry, you could do 60 measurements in 1-2 days fairly easily. And so overall, our aim is to reduce the overall time for an MDRS-based IVBE. So how did we go about that? Well, firstly, we sourced some Nasonex, which is the RLD for memetosome furoate, and then we developed what you might call a typical MDRS method to measure the API particle size. And so here we have some of the method details. Again, I'm not going to go through all of them in detail, just more if you want to look at them later. But with that method, you would measure a PSD looking something like that. I will, however, draw your attention to, we applied a 2-micron diameter filter to the analysis. So you might be able to see if I get my point. So here, we're not measuring any particles smaller than 2 microns. And the reason for this was the Raman laser itself is 2 microns in diameter. And so if you measure particles smaller than that, you are the accuracy of the chemical assignment gets lower and lower the smaller and smaller you get. And we also saw during the method development that there was a relatively small proportion of those particles in the whole sample were less than 2 microns anyway. We also applied some convexity, solidity, and elongation filters, which are morphological filters during the microscopy section, which are fairly standard parameters. We did note that the PSD you measure with the microscopy pre-Raman identification, so this is without the selectivity element, was still relatively similar to the measurement you get post-Raman ID from MDRS. However, there is an intrinsic error in that measurement because you haven't yet removed the excipients from the analysis. Remember, the aim is to measure a PSD of just the API within that suspension. And so to take it further, to optimize it, you can do some sample-specific tailoring of the morphological filters. And that will more closely align the PSDs, both pre and post-Raman ID. And so to do that, we built some known sets of API and non-API particles from the preliminary Raman data. And we overlaid those 2 sets for a whole host of morphological filters to see if we could further differentiate API from excipient. And the best case we saw was with a parameter called intensity mean. And that is essentially a measure of the brightness of a particle when it's imaged under the microscope. And so we can see in these examples here with the API in the red is on average darker when you see it compared to the excipient. It is a subtle difference, and you can see those populations overlap. However, they are separated enough so that you can cut out a significant proportion of the excipient particles whilst keeping all of the API population. And so we added an intensity mean filter to the analysis, creating what we're going to call an optimized microscopy method. And so then the PSDs you get pre and post Raman ID now very closely align. And so these are all from the same sample we measure, and we can see that the typical microscopy is the clear outlier compared to the optimized microscopy and the MDRS. Again, it's a subtle difference, and we did have to zoom in, but we will see that it is a relevant difference. And so we took 10 samples from the same batch, and we performed all of these 3 tests on each of the 10 samples to compare the results. And so we can see here that the average particle size, so on the left-hand column for the DN50 mean, for the average optimized microscopy results, they were matching the MDRS. We're taking the MDRS essentially as the gold standard measurement within the variation there. However, they didn't match the typical microscopy, so that intensity mean filter was necessary to be able to get measurements which agreed with each other. We then did an exercise where we sequentially processed down the scan area for our measurement to see how quick we could make it. And we saw that with microscopy, you can image sufficient particles to build a reliable PSD in a matter of minutes, whereas MDRS takes hours because you have to take all of those Raman spectra. And so even though we have, for the smallest measurements, still only a couple of hours, that is only for spectral acquisition. When you add in sample preparation, instrument calibration, etc., you're still only talking one sample a day. Whereas for microscopy, you can do multiple samples per day. So the question is, if matching PSDs can be acquired with the two methods, why are we still performing full MDRS for every replicate? And so on the left-hand side, in the blue, we've got the results for the optimized microscopy. On the right-hand side is a combined situation where we've taken the first 5 replicates from the optimized microscopy and the first 5 from the MDRS in yellow. And so it's a situation where within the 10 replicates per batch for your IVBE, you do a subset with MDRS and then the remainder with microscopy. And you can see the results match with each other quite well. And so our proposal is, what you now do for an MDRS-based IVBE is you develop a very sufficiently optimized microscopy method, use the Raman ID to prove that those PSDs match each other pre and post. Then during your IVBE, you analyze a subset of the 10 reps per batch with MDRS, and then the remainder made up from the faster microscopy. And so the advantage is overall you're reducing the whole time of your study. The amount of time reduction depends on the MDRS to microscopy balance, which would have to be determined statistically for each product, but we would still expect a lot of reduction. And so these results are promising. However, admittedly, this was a single batch of a single product, so it's merely a proof of concept study. So the next step is clearly to look at new batches and new products, which we've already started doing, and look at some deeper statistical analysis. If you did encounter a situation where a product wouldn't be able to be analysed using this regime, the worst case scenario is you can still use the conventionally accepted study regime of everything with MDRS, so it can only really get better. We do also need to look at some orthogonal techniques to look at that fine particle fraction, which we cut out originally. As an example, laser diffraction has been mentioned by the regulatory guidance, and eventually we would need the regulators to give a hypothetical acceptance of this regime, because clearly no manufacturer is going to pay for a study like this without knowing that they will say yes to it should their results be sufficient. But overall, we believe this work shows great promise and has a huge potential for cost and time savings. So thank you. We've also brought a poster and a stand with people on it if you want to go and know more, or you can get in touch with there. So thank you. I'm happy to take some questions.